Learning Objective 10 talks about pain modulation and pain amplification. I want to begin with differentiating at a high level pain modulating systems from pain amplification systems. The pain modulating systems are over here on the left side of this screen, and they are essentially systems that are inborn within us that have the ability for us to kind of manage our own pain intensities. And these two systems are known as the gate control theory of pain and descending inhibition. They essentially improve your overall pain state. Pain amplification, over here on the right, takes place because of two kind of theories, if you will, of how this can come about. And one is known as peripheral and the other is known as central sensitization. So this is essentially sensitization of the nervous system, either taking place in neurons of the CNS or in nerves of the PNS, the peripheral nervous system and central nervous system. And when they are pathological processes, when these pathological processes are ongoing, they worsen or amplify pain. First, we're going to talk about those pain modulating systems. The first is what was called the gate control theory of pain. This was first published in 1965 by two very famous researchers known as Melzack and Wall. This theory is essentially about when you have a pain signal, you can interrupt or diminish that pain signal using a competing signal for the same input. And this is essentially if you fall down and you hit your elbow, you rub your elbow on the spot where it hurts to ease the pain. The second is what we call descending inhibition. Descending inhibition is more about dampening, or you could even say putting a brake on the pain signal on its way that's coming up from the periphery up to the cortex. And there's a very specific type of descending inhibition known as presynaptic inhibition that we will also talk about. So to compare and contrast these, gate control theory of pain is essentially more about you have a pain signal coming in from the periphery to the brain stem, the spinal cord, or the CNS, and there is an interruption. So there's an interruption in the signal coming up. Descending inhibition is more about a signal coming down from the brain, particularly the brain stem, and that signal is coming down to meet the pain signals and interrupts the signal before it gets up to the brain. So I hope that makes sense. That gate control theory is more about interrupting an incoming signal. Descending inhibition is more about diminishing a signal from the top down. Let's begin with the gate control theory. In order to talk about what the gate control theory is and how it actually works, let's look at just kind of a baseline neutral activity in the dorsal horn. So this is essentially a graphic that's representing no pain. This is the absence of input from C fibers. There's just nothing going on. The person is totally fine, no pain at all. So let's look at this graphic. What we see here on the left is a C fiber. That is that slow, dull, throbbing pain. C fibers have central branches onto which they will synapse onto a secondary sensory neuron that will ascend on up to the brain. Now they also have a variety of other branches that would kind of split off from the main trunk, if you will, of the C fiber and go to others. And one of the synapses that's formed is something called an inhibitory interneuron. So if this is your primary sensory afferent, this is the secondary sensory afferent, you have other cells, other neurons just kind of sprinkled in all over the place. And their essentially job is to inhibit everything they touch. And they are called interneurons because they kind of squeeze in between functions of two of the bigger neuron pathways. So in this case, you have an inhibitory interneuron that is essentially tonically active. That means it's just spontaneously active all the time, and it's inhibitory because it releases an inhibitory neurotransmitter, glycine in this instance. Glycine is released from its presynaptic side, crosses synaptic cleft, and then is going to get taken up on the postsynaptic side, but since it's inhibitory, of course, this secondary sensory neuron is also deactivated because not only is there an inhibitory interneuron acting on it, there's no activity coming in from the C fiber because we're not in any pain. So in this case, if this inhibitory interneuron is functioning, it's kind of like the gate. The gate is closed here. There is no getting into the backyard, you could say. Now, I want you to focus first on this left hand side of this figure, this left hand box. We're going to now introduce pain. We're going to say that the person has smashed their thumb with a hammer really bad and they have a sudden, strong, painful stimulus. Now you would imagine that that noxious sensation is going to be transmitted up through the C fiber. That C fiber is very activated now. That C fiber, if you think back to our previous section, what kind of neurotransmitters does it release? Well, it releases excitatory amino acids, such as glutamate, and neuropeptides into the synaptic cleft. It gets taken up on the postsynaptic side by the secondary sensory neuron. And then this secondary sensory neuron is activated and it sends on up through the chain a strong painful stimulus up to the brain. Okay, that makes sense. You're in pain, the brain knows you're in pain. Owie. Now, simultaneously, remember how I said that there are sometimes other little branches that kind of diverge off from the main C fiber? Well, due to mechanisms that I won't go into here, they can release actually inhibitory neurotransmitters. Now, please do not let that confuse you. When you see C fiber, I want you to think excitatory amino acids and as well a neuropeptide. Now this inhibition, the mechanism behind the way this little section here works is not very well known, but there is some signaling mechanism that says that because of this little side branch, there is a different little section to the C fiber and it synapses not on the secondary sensory neuron, but it synapses on the interneuron, that little inhibitory interneuron that we saw in the last slide. So if you inhibit an inhibitory interneuron, that means inhibition stops. Therefore, this secondary sensory afferent is able to transmit pain. We call this the gate has been opened. And the gate has been opened because this inhibitory interneuron swings open and you're able to get a signal passed on up into the brain. Okay? Now, where the gate control theory really comes into play is over here in this right-hand side figure. Now we have, if you look at this kind of lower section, your painful stimulus. It's the exact same pain. You smashed your thumb with a hammer and it hurts really bad and you have C fiber activity. Same situation is going on here. You have C fiber activity, releasing excitatory amino acids and neuropeptides from the presynaptic side, and it activates the postsynaptic side, which in this case is the secondary sensory afferent. Now, there's a lot of activity going on over here involving this little bitty interneuron. So what's going on? Now let's say you, at the same time you smash your thumb, you grab it with your other hand and you start shaking it and you're rubbing it and you're going, ow, 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 meaning you are inducing other stimuli. That's not normally painful. You're touching your thumb, maybe you kiss it, maybe you're shaking it, you're doing some sort of something to help relieve that pain. That information is encoded through a larger afferent fiber, in this case an A beta fiber. That afferent fiber comes in and it has a branch that also touches synapses with the exact same interneuron that we saw before. So this interneuron is a busy little interneuron. It's getting branches from the C fiber and now from the A beta fiber. But since the C fiber was kind of shutting it off, now we have a conflicting signal. We have an activating sort of activity, which is what this plus sign is denoting, coming from that A beta fiber. And as a result, there's kind of an in-between activation. It's light blue here because it's not fully activated all the way, but it's not all the way inactive. It's kind of half on, half off, if you will. And as a result, it is able to release a little bit of glycine, not as much as it was, but enough to induce a dampening effect onto this secondary sensory neuron. If you note here, this secondary sensory neuron is kind of a pale green now compared to the left side, where we had full-on pain, but nothing was happening with the interneuron. Now the interneuron has kind of applied the brakes, if you will. So if we were going 45 miles per hour, this interneuron has touched the brakes to slow the car down to, say, 25 miles per hour. So you still get a painful stimulus that is sent on up to the brain, but the intensity of that painful stimulus is decreased. And that, in and of itself, is the proposed mechanism behind the gait control theory, and a very simplified series of circuitry going on in the dorsal horn that explains it. Now, again, this is still theory. It's not like the theory of gravity where we absolutely all believe gravity is real. This is a theory. All of these mechanisms aren't worked out yet, but there is evidence for some of it. And most anecdotally, we've probably all done this very thing where you have something that hurts and you go to rub it or touch it or shake or something like that. And that, for some reason, helps decrease the intensity of the pain. Okay, so that's all I'm going to talk about with the gait control theory. Next, we're going to talk about something called presynaptic inhibition and then descending inhibition. Now, if you remember back to the very last part of the gait control theory slides, I showed you that interneuron, that inhibitory interneuron that's kind of squeezing its way in between the primary and the secondary sensory afferents in the nociceptive chain. Well, technically, I didn't get into which part of the synapse it's really working on. It can be working, an inhibitory signal can be working on the postsynaptic side, which is kind of what the gait control theory is describing. But there is also a presynaptic inhibition as well, meaning it can work on the presynaptic C fiber afferent that's coming in. It can work on that side of the synapse. And what that does is it's also another way to kind of put on the brakes to decrease the intensity of the signal before it moves on up to the brain. So the primary afferent neurotransmission, controlled by inhibitory mechanisms that affect the pre and the postsynaptic side of that important synapse in the dorsal horn. Presynaptic inhibition has been proposed as probably the more powerful form of inhibitory control, and therefore induces a big effect. Now, what is presynaptic inhibition really? Are you really inhibiting something? Well, not really. The name is actually kind of a misnomer. This presynaptic inhibition is actually more of a decrease of the excitatory signal coming in. So if you have an excitatory signal coming in that's coming in at 100, and then you decrease it by 25, that would take the signal down to 75. Okay, so say you have 100 signals coming in and then you take away 25, now you're down to 75. Well, what this is more like, instead of having a signal all the way up to 100, then you take it down to 75, it's actually more of the signal itself only reaches 75. Okay, so it's a diminished excitatory signal. Rather than having a very powerful signal that you have to retract, it's actually just diminished in the first place. And it does so because there is a hyperpolarization of the presynaptic side of the membrane. Now, I know this is probably throwing all of your ideas of how synaptic transmission works for a loop right now, because you think, well, shouldn't signal always travel from presynaptic to the cleft to the postsynaptic side? Well, it can get more complicated than that, and this is kind of a glimpse into that complexity. What that means is there can be an influx of chloride into that kind of presynaptic side terminal, a little bit higher up on the chain too, but really at that presynaptic side. And then there's a little bit of a hyperpolarization at that area. And what ends up happening is you can still have a transmission of some action potential, some neurotransmitter release. It's just not as much as it would have because the intensity of the calcium that's entering the cytosol simply just isn't as much. And this is part of that graded receptor response that we talk about when we really think about sensory afferents and in particular nociceptors. They have a graded response rather than a binary action potential, meaning the action potential is either there or not. It has a graded response. It can kind of become intense and less intense, and more intense and less intense. So that's essentially presynaptic inhibition. Next, we're going to talk about descending inhibition. And the reason why I had to talk about presynaptic inhibition first is that this is all going to come together in something called descending inhibition. All right, here we are at descending inhibition. Globally, descending inhibition means that you're going to have inhibition of activity in that tiny little circuitry in the dorsal horn involving nociception as it is arising from the higher brain center. So it's arising from areas in the brainstem. The signal comes down to the relevant level in the spinal cord, and it has an impact on the dorsal horn signaling circuitry. So let's start at kind of the highest levels and think about where does the signal originate? Well, it originally originates in the periaqueductal gray matter. Now you might ask yourself, well, if anything in the brain, does anything in the brain ever really originate anywhere? And the answer is no, not really. There's always something else that stimulates it. So you might ask, well, what stimulates the periaqueductal gray matter? Well, the periaqueductal gray matter are themselves activated by opiates, excitatory amino acids, and cannabinoids. And those come from various other brain regions that I won't get into right now. So we have these signals that activate the PAG. The PAG sends a branch on down through the spinal cord to one of two regions. It can go to the locus coeruleus or it can go to the raphe nucleus. If these projections go to the locus coeruleus, what happens here is it will activate a neuron cell body that's located in the locus coeruleus, and it does so by excitatory amino acid mechanisms. Now, this locus coeruleus neuron has a projection that will go on down to the relevant area in the dorsal horn. And upon reaching that area, it will release its neurotransmitter. The locus coeruleus located neurons release norepinephrine. So this is a noradrenergic projection pathway. Now let's start back up at the PAG again. The PAG has its neuron. It's been activated by opiates, excitatory amino acids, and cannabinoids. It sends a descending projection now, instead of the locus coeruleus, to the raphe nucleus, releases a excitatory amino acid, and activates a neuron cell body that's located in the raphe nucleus. This neuron cell body has a projection as well that goes down, in this case, down to the same level of the dorsal horn, and it releases its neurotransmitter, which is serotonin. So that means that we have a noradrenergic projection pathway originating from the locus coeruleus and a serotonergic projection pathway originating from the raphe nucleus. Both will reach the dorsal horn, both can inhibit, or both will activate inhibitory interneurons. Those inhibitory interneurons can release glycine, or in this case, opiates like enkephalin. Enkephalin, as a neurotransmitter, will activate its receptor, in this case, they are the mu receptors, on either the presynaptic side, we just got done talking about presynaptic inhibition, but it can also work on the postsynaptic side at that level that we just got done talking about, at the level in the dorsal horn where the incoming C fiber is about to synapse on its secondary sensory neuron. This results in a reduction of neurotransmitter and neuropeptide release from that central terminal of the C fiber. therefore reducing nociception. All right, I know that that was a lot, so let's now go to the next slide and look at a picture of what this circuitry can look like. All right, this is a picture of what that circuitry in the dorsal horn can look like. Here we have incoming projections from neurons originating where? The raphe nucleus and the locus ceruleus. Their projection terminals will come down and they will release either serotonin or norepinephrine into this area. Now, depending on what it is they are exactly synapsing on, if they're synapsing on an interneuron, it will activate that interneuron to release an inhibitory neurotransmitter. In this case, it's enkephalin. It can also release glycine. That interneuron will kind of squeeze its way into that either the presynaptic side or the postsynaptic side and induce a presynaptic or postsynaptic inhibition that will then impact that secondary sensory neuron and reduce the intensity of nociception that would otherwise go on to the brain. And if it isn't activating the interneuron, it can instead act directly on that secondary sensory neuron cell body itself. So here it's working at pre and postsynaptic inhibition. Here it's just straight out inhibiting that secondary sensory neuron cell body. Together, whether it's doing one or the other, it suppresses the activity of those spinothalamic projection neurons and therefore reduces the intensity of pain. Now you can imagine that if something were to go awry with this system, whether it's gate control theory, pre or postsynaptic inhibition, or descending inhibition, you can imagine that if you lose your your dampener of pain, if you lose the ability to suppress spinothalamic projection neurons, then that might result in an increased or hyperactive pain state. Now, speaking of hyperactive pain states, we're now going to talk about two types of sensitization that can occur and affect the nociceptor and nociceptive pathways. And they are central sensitization and peripheral sensitization. Very simply, central sensitization affects the activity at a synapse level that if in the central nervous system along the nerve pathways that transmit nociceptive signals. That can be in the spinal cord or it can be higher on up in the brain and in various brain regions that are responsible for interpreting nociception. It results in a hypersensitivity that together with a wide variety of cellular and molecular mechanisms, it really impacts the plasticity of the synapses themselves. Again, this can be at the level of the spinal cord, brain regions, or both. And essentially what happens is it reduces the threshold of the involved neurons to be activated by noxious stimuli, meaning it just takes less noxious stimuli to induce a big response. It involves synaptic plasticity, particularly persistent stimulation over and over and over again of excitatory amino acid receptors. If you know what those receptors are that bind those excitatory amino acids, they are the NMDA receptors, non-NMDA receptors, if you remember those. And then persistent stimulation through intracellular signaling cascades. This particularly involves what we call central inflammation, meaning that there are pro-inflammatory signals released by immune cells in the brain or the spinal cord, in this case can be from glial cells, that contribute to activation of the neurons in such a way that they become sensitized. So that's central sensitization in a nutshell. Next we'll look at peripheral sensitization. In contrast, peripheral sensitization involves those neuroplastic changes that relate to the function, the profile, the structure, receptor ion channel neurotransmission levels, all of that when it occurs in the peripheral nervous system. Then it is peripheral sensitization. The mechanism that contributes to peripheral sensitization is is attributed to essentially a continuous exposure of those peripheral afferents to an inflammatory soup. So essentially just bathing those free nerve endings in inflammatory mediators over and over and over again for a long period of time can sensitize the nociceptor. As a result, that can increase the intensity and duration of pain, and that inflammatory soup can come from nearby immune cells. So if you recall, because of those neuropeptides that can be released upon nociceptor activation, neuropeptides can be released back out into the tissues around the free nerve endings that they came from. Those neuropeptides can go back onto immune cells, recruit more, and then set up a series of neuroimmune activation. And it sets up this sort of cycle that, if you don't interrupt, can lead to eventually peripheral sensitization. The most famous inflammatory mediators that are involved in doing this are the prostaglandins, prostaglandin E2 in particular, PGE2, and bradykinin. Eventually, it reduces the firing threshold, increases nociceptor responsiveness, and it can do so to such a state that, if you remember that TRPV1 receptor, how TRPV1 can be activated by noxious heat well over 50 degrees Celsius. Well, what can end up happening is that you can become so sensitized that even at normal body temperature, 98 degrees Fahrenheit, you can activate TRP receptors. And if you're activating TRP receptors, particularly TRPV1, you're going to get induction of nociception and probably pain as a result. So both central and peripheral sensitization can lead to chronic pain states that are very complex molecularly and mechanistically, and are very difficult to treat as a result of that. This table is a great summary of the compare and contrast, or the differences when it comes to the location, what's affected, the underlying mechanism, and the outcome related to central and peripheral sensitization. The question on everybody's mind is, how does chronic pain develop? Does chronic pain develop because there is a problem in the periphery, such as peripheral sensitization? Is there a problem in the central nervous system, such that there is central sensitization? Or maybe it's something else. Maybe there is a problem with our pain modulating systems. Those modulating systems are the gate control theory of pain and descending inhibition. Is there a problem with these systems? And why does chronic pain develop in some people, but not everyone? And what are the initiating factors? And finally, how do we treat and interrupt this cycle of chronic pain? Well, this is really the topic for all researchers who work in the pain field, myself included. And the answer is, we just don't really know. It's probably everything, such that, an individual can have all problems with all of these systems. They can have peripheral sensitization, they can have central sensitization, and deficient modulating systems, or it could just be one of the three, or it could be none of them, and there is something else completely otherwise going on that we haven't discovered yet. So I hope that you found this information to be very interesting, and stay tuned. Hopefully there remains lots to discover and lots to understand in order to help your future patients, especially those in chronic pain. The last learning objective is about the highest level of pain processing and the interpretation of painful events. Now, you may recall from earlier in this lecture series, we talked a lot about the primary and secondary, and then the association cortices involved in the somatosenses. When it comes to pain, really S1 and S2, they do receive input from nociceptors, but they mostly play a role in the crude localization of pain. More importantly is the insular cortex. The insular cortex is important in the interpretation of nociception, particularly as an unpleasant and experience that we want to not experience again. The insular cortex not only processes information about the internal state of the body, it can contribute to that physiologic autonomic response to pain, It does a lot of integration of signals that are related to pain, and damage causes a syndrome called asymbolia. I'm not going to go into that here, and I'm not going to hold you responsible for that, but there's a hyperlink to it if you are interested in learning more. So most importantly is insular cortex in its ability to tell you that nociception or pain is unpleasant. Other areas involved in pain processing is the amygdala. The amygdala is important to the emotional component of pain. So, for instance, getting angry, getting sad, more of that. More the insular cortex is more about pain is unpleasant, I don't like it. The amygdala is more about the emotional response to pain. And then finally, when you have pain that is arising from the viscera or visceral input, that's going to begin to integrate the physiologic autonomic response. Then we're talking about the hypothalamus and the medulla working together. Just remember that a lesion in any single area may change the experience of pain, but it does not abolish it completely. And that concludes this lecture series. As always, please be sure to reach out to me if you have any questions. Thank you.